Herein, two different methods for evaluating sperm chemoattraction are described. For the two-chamber assay, activated frog sperm are first dispensed into inserts with porous membrane bottoms set into the wells of a 24-well plate. Then, once the sperm are in place, a chemoattractant is carefully added to the bottom of the well to initiate a chemoattractive gradient. Next, the sperm are incubated to allow the cells time to cross the porous membrane in response to the chemotactic gradient. Finally, the insert is removed, and the sperm that have migrated to the bottom of the well in the two-chamber assay are enumerated by hemocytometer. Ultimately, the activity of sperm chemoattractants can be compared and quantified, for example, by a dose-response curve analysis. For the Siegmund chamber tracking assay, a cover glass is first applied to the Siegmund chamber with silicone oil and placed upside down over the circular opening of a microscope stage. One side of the trough of the chamber is then filled with activated frog sperm. Next, the other trough is filled with the chemoattractant solution. After 5 minutes, the chemotactic gradient forms, and at this time, the sperm can be videotaped swimming along the gradient. Ultimately, the footage acquired from the Siegmund chamber tracking assay can be analyzed to determine the velocity of sperm and the chemokinetic patterns and trajectories of sperm travel in response to a chemoattractant gradient.
These two methods for detecting sperm chemotaxis are complementary, and they produce different types of data. The two-chamber assay allows sperm to pass through a porous membrane, thereby allowing quantitation of the sperm response to a chemoattractant.